Welcome to the world of bioinformatics, where code meets life and research finds its future. This is Biotech-On.
Hey everyone, and welcome back to another explainer. If you're a biology student, get ready, because today we are going to completely rethink everything we thought we knew about protein biology. You know, for decades, the central dogma of structural biology has been pretty rigid, pun definitely intended. But today, we're throwing out the script. We're going to explore this hidden world of tiny, incredibly dynamic molecules that are actually pulling the strings behind some of your cell's most absolutely vital processes. We are talking about intrinsically disordered microproteins. To kick things off, I need you to do a little bit of unlearning with me. Think about this. Does structure always equal function? From literally day one of your biochem classes, you've been taught that classic lock and key paradigm, right? You're told a protein has to fold into this highly specific, super stable, three-dimensional shape just to do its job. But what if I told you that a lack of structure might actually be a superpower? What if being a shapeshifter lets a protein pull off things that a rigid, folded protein just can't do? So to really get this, we have to talk about intrinsically disordered proteins, or IDPs. To put it in the easiest way possible, I want you to imagine a protein not as a solid little puzzle piece, but as a dynamic, flexible cloud of amino acids. These proteins, or at least really large regions of them, simply don't adopt a single, stable 3D shape when they're on their own. Instead, they're constantly fluctuating. They are the ultimate molecular shapeshifters. And as you'll see in a minute, when you combine this intrinsic disorder with a really small size, you end up with microproteins that have some truly mind-blowing biological capabilities. Okay, so here's our game plan for today. First, we'll officially meet these shape-shifting microproteins. Then, we'll dive into why their flexibility is such a superpower, look at some master regulators in action, break down their phosphorylation on-off switch, and finally, we'll tackle the massive challenge of drugging the undruggable. All right, let's jump right into section 1. Meet the shape-shifting microproteins, which were basically hiding in our junk DNA. For the longest time, huge regions of our genome were just written off as non-coding junk. But thanks to some wild advances in ribosome profiling, we've actually discovered thousands of small open reading frames, or SMARFs, that are busy producing these dynamic little microproteins. And what's wild is that these tiny disordered microproteins operate everywhere. They're across incredibly diverse cellular domains. They aren't just stuck doing one niche job in the corner of the cell. They're heavily involved in DNA-dependent processes like genomic repair. They help form biomolecular condensates. They manage cellular homeostasis right at the membrane, and they are tightly controlled by phosphorylation. They're basically everywhere, acting as these vital adapters and regulators for the larger, more traditional protein complexes we're used to studying. Which brings us to section 2, flexibility as a superpower. Let's look at the biophysics of disorder, because we really need to understand how exactly a protein's physical flexibility dictates its role in the cell. Let's do a quick comparison. Think about the classic canonical proteins you usually study versus our disordered microproteins. Canonical proteins are kind of like a Swiss Army knife. You know, they're rigid, they have these stable, well-defined binding pockets, and they generally exist in just one single conformation. Disordered microproteins, they're more like a flexible piece of string with sticky spots all over it. Because they exist as these dynamic ensembles, they can literally wrap around their targets. This allows for multivalent interactions, meaning they can bind to multiple different partners, sometimes all at exactly the same time, which makes them incredibly versatile. But you might be wondering, why are they so flexible in the first place? Well, it all comes down to the recipe. When researchers looked at the sequences of these microproteins, they found a very specific amino acid signature. They are highly enriched in disorder-promoting amino acids. We're talking alanine, glycine, proline, and arginine. Proline, for example, is notorious for breaking up rigid alpha helices. And at the exact same time, they are totally depleted in those bulky, order-promoting residues like isoleucine and tyrosine. So this specific biochemical recipe, that's the fundamental secret behind their shapeshifting. Moving on to section 3, let's see these master regulators of the cell in action. Let's actually walk through a few real-world examples here. First up, we have siren. Siren interacts with the Cu70/Cu80 complex, and its job is to regulate DNA double-strand break repair. It literally protects your DNA by inhibiting classical non-homologous end joining during specific cell cycle phases, which ensures the repair is error-free. Next, we've got NBDY, which stands for nobody. This little microprotein interacts with decapping complexes to regulate P-body assembly.
absolutely crucial for managing RNA turnover. And finally, PLN, or phospholambin. This one is a master regulator in your cardiac muscle cells. It binds to the calcium pump circa in the sarcoplasmic reticulum to maintain calcium homeostasis. So yeah, these tiny, flexible proteins are practically running the show.
And the way they run the show is super fascinating, especially when we talk about liquid-liquid phase separation, or LLPS. Think of LLPS kind of like oil separating from water in a vinaigrette. Inside the cell, this process creates biomolecular condensates, which are these organelle-like droplets that don't even have membranes. Because microproteins like NBDY and others like P14RF are so highly flexible and have all these multiple interacting points, they act as the ultimate molecular glue. They use multivalent electrostatic interactions to pull specific RNAs and proteins together, and that drives the phase separation that forms structures like P-bodies or nucleoli.
Okay, section 4, the phosphorylation on-off switch. Here's a really fun question. If these proteins are just flexible, shape-shifting clouds, how on earth does the cell actually control them? Like, how do you turn a cloud on and off?
The answer is actually this beautifully elegant regulatory mechanism, post-translational modification, mainly phosphorylation. It works in 3 steps. Step 1. A kinase enzyme comes along and slaps a negatively charged phosphate group right onto the microprotein. Step 2, this completely shifts the local charge distribution of our little dynamic cloud. And step 3, that charge shift physically forces the protein to change its shape. Sometimes it transitions from a disordered state into a structured, ordered state when it binds its target, or sometimes the exact reverse happens. This biophysical switch totally alters the microprotein's ability to bind to its partners.
To put that into perspective, let's see how this switch plays out in reality. When NBDY gets phosphorylated, that charge shift actually drives the dissociation of those P-body condensates we just talked about. It literally dissolves the molecular glue. Boom, gone. With PLN, phosphorylation induces a conformational change that stops it from inhibiting the circa calcium pump, which is exactly what allows your heart muscle to relax properly between beats. Pretty important, right? And for the pro-apoptotic microprotein NOXA, phosphorylation physically restricts its shape through internal electrostatic interactions. This completely prevents it from binding to the survival protein MCL1, which effectively stops it from triggering cell death. All right, our final section, number 5, drugging the undruggable. This is the big therapeutics climax. Think about it. How do you design a traditional, rigid drug for a protein that is literally a dynamic, shape-shifting cloud? Well, modern chemical biology is fighting back against this undruggable label using some incredibly clever targeting strategies. First off, we've got nanobody-based degraders. Researchers are engineering these highly specific nanobodies that can grab onto a disordered region and just drag the entire protein straight to the cell's trash can, the proteasome, to be degraded. Second, there's covalent ligand discovery. For instance, researchers found a molecule called EN4 that covalently binds to a highly reactive, hidden cysteine residue right inside the disordered region of the notorious cancer protein MYC, completely disrupting its function. And finally, using chemoproteomics, scientists are mapping entire networks of reactive arginines within these flexible clouds. This paves the way to target the exact residues that drive phase separation. We are literally figuring out how to drug the cloud. Which brings us to my final thought for today. We have gone from dismissing vast stretches of our genome as just useless junk to discovering this vibrant, dynamic universe of intrinsically disordered microproteins that are regulating the absolute core functions of our cells. We are sitting at the absolute frontier of proteomics right now. So I want to leave you with this. Considering we've only functionally characterized a tiny, tiny fraction of these translated smorphs, what other shape-shifting, life-saving microproteins are out there hiding in plain sight, just waiting for you to discover them? Think about that as you head into your next lab session. Thanks for hanging out with me on The Explainer, and as always, keep questioning everything.